Hello, welcome back to Coffee Break Science, brought to you by the NIHR BRC Devices and Advanced Therapies theme. I'm your host, Arsena Stola. Today we're diving into the human genome, the entire set of instructions found within the nucleus of a cell that contains the code to allow us to function. And today we're going to dive into what happens to the genome when cancer forms and how the cancer genome and the mutational signatures that are etched at an individual level can help us inform treatment for patients. Today, I am delighted to be joined by Professor Serena Nik-Zainal, who is incredibly inspiring. She's an NIHR Research Professor of Genomic Medicine and Bioinformatics at the Early Cancer Institute, University of Cambridge, and an honorary consultant in clinical genetics at Addenbrooke's Hospital. She was trained in medicine at the University of Cambridge with a scholarship funded by Petronas, and completed her PhD at the Wellcome Sanger Institute in 2009, where she explored breast cancer using whole genome sequencing and has won numerous prestigious awards in recognition of her groundbreaking work. So yeah, I'm very humbled that you accepted my invitation. Thank you so much.
Thank you, Asen, for the invitation to come into the podcast. I'm feeling really, really honored to be here and also, humbled myself to be in your presence, really. When I first met you and you told me about your story, I thought, oh, what a courageous young lady. So I'm more than delighted to be here today. Thank you so much.
Thank you. So I did a bit of stalking, as one does, and saw that you are a self-confessed nerd who went from being a clinician who treated kids who had inherited rare disorders to being at the very forefront of genomic research here in Cambridge, which is really revolutionizing patient care. I myself have personally benefited from genomic sequencing and research. So can you walk us through this journey, how you went from being a qualified clinician to leading all this amazing research?
Oh, thank you for the question. So I'm from Malaysia originally and came to the UK, very, very privileged to have won a scholarship from Petronas. And I really, my family wanted me to be a doctor. We come from that kind of background where you're going to be doctors, lawyers, or engineers. But, I did have a passion for medicine and I started doing medicine here. And I suppose one of the things that my father instilled in me, the day I left, the day he put me on an aeroplane on the scholarship, he said, your opportunities are over there. And so, if you see an opportunity, make sure you grab it and run with it, right? And don't worry about what you're hearing around you and what people are trying to say. So coming to Cambridge was an enormous opportunity. And, Cambridge is such a hothouse of intelligence and, and education and sort of people who are experts in their field. So opportunities abound here, right? So I did medicine in Cambridge, and that was fun, that was inspiring. I kind of looked up to the people who were my seniors, the consultants who were here, and wanted to be like them. And I think one of the things that drives me is looking up at people and sort of going, okay, that's kind of what I want to be. That's who I want to be. And so I was very happy doing medicine, and I thought I was going to be a full-on NHS consultant doctor. And in those days, becoming a consultant in Cambridge, my husband was a consultant here already, so becoming a consultant in Cambridge would have required me to have had a PhD. So I trained in clinical genetics, which is a specialty of rare genetic disorders. And we're constantly trying to diagnose children with different abnormalities, adults too. And I think our sort of hit rate, our diagnostic rate was about 10%, so it was pretty poor. But new technologies were coming. So next generation sequencing started to come in the late 2000s. I was still a trainee, I was still a registrar at that point, specializing in my training in clinical genetics. And I remember SNP arrays started coming in. And I remember sitting with the result with a family and sort of looking at the result going, I'm not sure I fully understand this result. And I am the doctor who has to explain this to the family. And if I don't fully understand it, how are they going to understand it? And that was the point that I decided I did want to do a PhD in technology. So being in Cambridge, and I remember my father, in fact, it was the day before my father died. He said to me, if you want to do technology, there's that big institute down the road called the Sanger Institute. It's been involved in the Human Genome Project. You got to go there and do your training there for your PhD. So I think that was a Sunday evening that I spoke to my dad. Monday, unfortunately, he died. And I was on maternity leave. And I remember thinking, okay, I'm going to go and have a look at the Sanger Institute. So I did. Life is full of serendipitous events. I met somebody who was inspiring me at the time there. And so again, this point of, being inspired by somebody comes in. So then I decided to do my PhD at the Sanger. And I probably couldn't give you good career advice because my life has always been sort of directed by life events and people around me. So when I went there and I did my PhD, I just found it super interesting. And I decided at that point, okay, actually, maybe I want to do this for a bit more than just a PhD. And that's how I started going into the research of cancer genome. So I got introduced at the Sanger Institute, just found it so interesting. And, I was so keen to just mess around with data all the time. This is when I figured out I was a nerd. And so I've stayed. And, that's the story of how it went. It wasn't particularly well planned or I can't say I was one of those people who thought I'm going to be an academic. I absolutely didn't think that. But I have been inspired in my journey all the way along by various people and by various events that have happened. Right place, right time.
Yeah, amazing. But you did all of that while you had a baby as well.
I had two.
Oh my goodness. Wow.
So I started my PhD with a 1-year-old and a 4-year-old.
Wow.
And I have to say, I didn't think too much about it. I just thought, I want to do a PhD. And okay, I've got 2 kids. And, but this is an opportunity. It was hard to get a PhD in those days. I was 34. I was not young. I was a mum of 2. I didn't have a pedigree behind me. I hadn't been to lots of big labs and had a glossy CV behind me. But I was given an opportunity by the Wellcome PhD program in Cambridge. I guess I had worked with some of those academic consultants as a junior doctor before, and I think they saw, that there was some potential in me, hopefully.
Clearly.
So that was an opportunity. That was I think it was a gamble for them, but it was an opportunity for me, and I wasn't going to waste it. I thought, well, I've got this chance now. It's been really hard to get a PhD at all.
Yeah.
So I thought, okay, I'm gonna do this. I'm gonna do this properly. It was hard, but yeah, it's doable. It is doable. Yeah.
And you got amazing publications from it, including that one that went on the cover of Cell, which is amazing.
Well, everybody sees the final product, right?
Yeah.
They go, wow, what an amazing final outcome. And it was an amazing final outcome. But there were many moments throughout that PhD, which all of us experience. And that's normal.
Yeah.
That you get sat down by your boss and he goes, or he or she goes, you haven't got any data, you're not making progress fast enough, And I think that's normal and we should normalize that. It is okay to be in that position sometimes during your PhD or during your postdoc years. But it is possible to accelerate and then to push really hard. I was in a team. There was a group, and as ever with any publication of any sort these days, you're often in a big team of people. And it's sort of knowing who you can work with and push things along that makes something come out at the other end. So it went well in the end.
Yeah. And all the way to now leading a group at the university. So how was that also just following a series of opportunities?
Yeah, I suppose my supervisors at the Sanger, they were quite realistic. They sort of said, well, if you want to be an academic medic, you're going to have to find your own funding and you're going to have to sort of support yourself because you sort of expect, if you expect sort of funding to come to you, it's not going to happen. So I think that was instilled in me at a very early stage and from the get-go. So I finished my PhD. I had to go back to the NHS to finish off my training, but I only had 6 months. So I was going to come out the other side, finished training, no job potentially, and I sort of had to figured this out fast. And I applied for an intermediate fellowship. I got that one. That was from the Wellcome. That allowed me to do an intermediate fellowship. And I did that believing I was going to do those 5 years and see how it went. And, there are hurdles. I went briefly to the LMB. I was going to work with Michael Neuberger there. Unfortunately, was diagnosed with cancer at the time. So I wasn't there for very long. He was quite sick. So I came away from the LMB and went back to the Sanger. So these things happen, don't they? You can plan things to the best of your abilities, but sometimes there are hurdles that come in your way. I was already working with Michael Neuberger prior to arriving there, so I did already learn quite a lot about how molecular biologists think. That was quite informative for me. I think that really was quite an important moment for me to sort of realize it wasn't enough for me to do computational work. So during my intermediate fellowship, I started to do some experimental work as well as the computational work alongside.
Okay.
And that was fun too. That was different. That was hard in a different way. That was fun too. And then I moved from the Sanger to Cambridge. I wanted to do more clinical stuff. I wanted to get things to patients. You know, my mom's like a typical Asian mom. She was like, you should just be a consultant in the NHS. Why are you a scientist? She couldn't get her head around it. She was like, you're not earning enough money. You should just be a consultant. She wanted to be able to say this. When I had that cover of Cell, Mom goes, I said, Mom, it's a nice publication and it's on the cover of the journal. And she's like, is that like a magazine? Like Vogue or Tatler, And I'm like, well, Cell's a major magazine, a major journal, right? I said, I'm on the cover. And she goes, is it your photograph? So I'm like, no, Mom, it's a picture of my science. Not impressed. She was not impressed. And it's really useful to have people like that for whom Nature, Cell and Science papers don't matter. And actually, her question to me was, how does this benefit mankind? That was just her brutal sort of, why is this useful? And so going to Cambridge, I did want to make sure that, these things that we found in the cancer genomes, we could use for clinical purposes, mutational signatures. And so that next phase was experimental work and then developing clinical algorithms to try to get to patients, and then working with a lot of people outside the Sanger and around the world, around the UK, to try to get things to patients. So it's been a super interesting journey. I can't say it was particularly well planned, but that's how it's gone.
Super cool. So that leads us really nicely onto my next question, which is, can you talk us through what the cancer genome is and how we can use these mutational signatures that you've mentioned, which are etched into an individual's genome? How can we use those to inform treatment for patients?
Sure. So, we all start off life as a single cell that you've inherited your genetic material from your mom and from your dad, 50% each. And then that cell's got to divide thousands of times to form the human that we are today. And a cancer will have developed from one of those cells and has just acquired a huge amount of mutations. So it has changed so dramatically. And with the advent of these new technologies, new sequencing technologies to read the human genome, we can now see every mutation that's in each person's cancer. And what we saw during the PhD was that, we only sequenced 21 women. It wasn't even a huge number of patients. At that time, it was a huge number, but today you'd say that's not a big number. But I remember first looking at the data and realizing, gosh, every woman has got a very different genome. Every cancer genome is different. We all get the same treatment, but the genomes are so different. They're so biologically different. And they had these patterns in them, which we call mutational signatures. And a lot of the work that started at the Sanger was about the mathematics of trying to define these signatures and trying to quantify them so we could see what signatures they were, and then we could also see how much each person had in their genome. So for example, if you have a lung cancer, we can see the impact of tobacco smoke on the lung cancer. Or if you have a skin cancer, you can see the impact of UV light on the skin cancer. That is a mutational process, a mutational signature. And I can even say how much of that signature is there. So I can see if someone's been a heavy smoker or not. So that was really powerful. And actually, it was during that PhD, we had 21 patients. And I remember, because I'm a clinical geneticist, so I deal with families with rare genetic disorders, and BRCA1, BRCA2 patients are some of them that we see in our general clinics. And I remember saying to my supervisor at the time that I think it'll be important for us to sequence some of BRCA1 and BRCA2 patients, given that that's my background, and BRCA1 and BRCA2 are genes that are involved in fixing DNA damage that happens to the DNA. So I thought, if you have that not functioning, you're going to see mutations. I'm so glad we did that, because otherwise I might not have had much to say in my PhD. But the moment of realizing that there was clinical power in that was, we knew that we had recruited 8 patients who were 4 BRCA1, 4 BRCA2. But there was a 9th patient whose tumor looked like it had BRCA1 signatures. And she was 32 years old. She was a young woman. And I remember thinking, gosh, this really looks like a BRCA1 cancer, but she has got no mutation. She's not known to have a mutation. And I remember looking back with one of my colleagues. We went back through her genetic material, her germline genome, and worked really hard, but we found it. We actually found a BRCA1 mutation in that young lady. And so we realized that the signatures could help inform a really important germline genetic factor in this patient and her family. So that was a very significant moment for us, I think. We didn't put that in the paper, but from an intellectual perspective and from a clinical perspective, that was a real moment of realization for us. So we sort of use BRCA1, BRCA2 as an example of a genetic disorder that has signatures. There's many other ones. Mismatch repair deficiency also has signatures. As I mentioned before, UV light and tobacco. So we can see the mutational damage that happens in each person's cancer, regardless of whether it's from the external environment or from the internal sort of cellular system. And that's important because we might be able to do something about it, right? We might be able to treat it more effectively or provide advice to people about how to avoid public health sort of interventions to sort of avoid some of these exposures.
So in your PhD work at the Sanger, you discovered a mechanistic insight using this whole genome sequencing technology called CATIGIS. Can you tell us what this mutational pattern has told us since your publication in Cell over 10 years ago?
So CATIGIS was something that we discovered by asking a very simple question, which is, are the mutations in your cancer genome, are they equally spaced out? Because your genome is huge, right? 3,000 million base pairs. And if I have 3,000 mutations and they're just distributed through the genome, are they sort of evenly spaced, roughly, right? So if you have 3,000 mutations and your genome is 3,000 million bases, then you expect a mutation every million bases or so. So we just asked a simple question, are they evenly spaced? And we found that most of the time they were, but sometimes there were areas where you did get many mutations that were really close together. And it was just almost a stupid question, the power of asking stupid questions. But we found that there were these strips where in our 21 patients that had these sort of lots of mutations close together, they looked like, and we presented it in a way that it looked like they were rainfall patterns. So it got called CATIGIS, which means storm of mutation. And here's the power of, we sort of talk about bioinformatics and computational genomics and data science. And yes, I use that to find those strips. But biology requires just looking at the data and playing around with it. And so when we looked at those mutations in great detail, sort of looking at the actual individual DNA molecules, the reads, the BAM files, we realized the mutations were always of a particular type. They were always C to T mutations, cytosine to thymine mutations. And then there was this moment where we're looking at the reads going, actually every C is always preceded by a T. So not just were they C to Ts, the Cs that were mutated always had an upstream or 5' T. That was weird. So you could see all these beautiful strips of red or black mutations next to a T. And then, I just did a bit of reading about what that could be, and I had to turn to molecular biology papers for that. And then we discovered that there was a family of enzymes called ApoBex that sometimes deaminated or was thought to deaminate cytosines to uracils and then to thymines. Then it becomes a thymine at the end of the process of cell division. And this is one of the things about being in Cambridge. The world expert in ApoBex was Michael Neuberger, the LMP. So I was like, oh, that's handy. We can just pick up the phone and call him. And so that's exactly what we did. Picked up the phone, called him, asked him what he thought. And I remember how excited he was to see this. I went to his lab, I presented it there, and they were just so excited. They thought it was ApoBex. They are, of course, the experts in it. So they could explain the mutational mechanism, like how the mutations arise. And then we did a set of experiments in yeast. And they partnered with his team and his lab to do those experiments to show that ApoBex were actually causing those strips of CATIGIS. So, yeah, it was serendipitous, like everything else. Right place, right time. But also just being able to ask stupid questions and not being afraid to ask them, I think. Yeah.
Oh, amazing. More recently, you and your team have been using machine learning methods to build an algorithm, two algorithms that I could find online called HR Detect, so homologous recombination detect, and MMR detect, so mismatch repair detect, I think. Can you tell us about how these tools were born and what the clinical applications are and whether they have been implemented or what the road kind of looks like?
So this is a great question, Asin. So, where did it come from? It came from sort of my mom going, how do you make this useful for mankind? And having a fantastic team of people who are prepared to explore using different methods. And AI and machine learning is like all the buzz, all the rage now. And while I do think there's a lot of value and use for them, I think you want to try to keep things as simple as possible. You don't want to give doctors black boxes. They don't like black boxes either. So we started with very simple models to try to see whether we could use signatures to predict whether somebody's got a BRCA cancer or not, because that was based on that 21 patients where we found that additional patient who had a BRCA1 mutation. So we said, okay, well, if we now have cancer genomes, if we could sequence lots of patients, and you design an algorithm to detect more people who have BRCA1 or BRCA2-like cancers, can you then identify more germline mutations or at least treat them with the same drugs that were created for the germline mutation carriers? Because we know about PARP inhibitors, we know about platinums that get given to patients with BRCA1, BRCA2 mutations. They are supposed to do better on these sorts of drugs. So can we identify additional patients who have BRCA1 and BRCA2-like cancers so that they could benefit from the same treatments? That was the premise. And we used very simple models and we made them a little bit more complex. And then we came up with this HR detect algorithm. And that was published 2017, I think it was. And actually it took me a couple of years to realize that, making algorithms is probably the easy bit. The hard bit is getting it to patients. At this point, I was already in Cambridge, and getting people to adopt algorithms to do the clinical studies to get to patients is hard. It's slow. And it doesn't get you many papers either. It's not valued necessarily in the same way. Be that as it may, you want to try to do it. So and again here, everything's collaborative, right? We ended up collaborating with people in Sweden, in London, and all sorts of places to go through that process of getting it to patients. And so I think it was by 2019, had worked with a team in Sweden to show that HR Detect could prognosticate. It could help to distinguish patients that were going to do well from the ones who were going to do badly. So if you had a HR Detect high tumor and you gave them the right treatments, they were the ones who were more likely to do well. And for the HR Detect low patients, these were patients that if you gave them standard of care, it made no difference. The outcomes were still poor. And that means it's not just important to find the HR Detect high patients and give them the PARP inhibitors and platinums. It's also important to identify HR Detect low and find new drugs or better ways of treating them more effectively. So that was 2019. And then by the time we got to 2020, 2021, we were starting to collaborate with clinical trialists to show that it could be predictive, so that we could use HR Detect to try to predict who might be sensitive to PARP inhibitors. So that's where we are now. We're trying to get it to clinical trials into patients.
Okay. Yeah, there's so much work that needs to go into doing any kind of implementation.
And a lot of people need to be involved, right? Not just people like us. We need to work with clinical trialists and, patient groups to sort of get the adoption of a new technology into the clinic. Yeah.
I remember speaking to one of your PhD students at the Personalised Breast Cancer Symposium who told me that she actually sometimes analyses some of the data from the Personalised Breast Cancer Programme, which is how my germline BRCA defect was detected. And we had this conversation, I wonder if you did the analysis. That I actually opened my letter this morning, my genetic letter, because I wanted to know exactly what technique had been used. Obviously, it's kind of you bury it and you kind of want to forget about it. But I actually realized it wasn't whole genome sequencing. I think it was a targeted twist panel, breast cancer susceptibility genes, so only 6 or 7 markers. And through that, they detected that BRCA1 was pathogenic. But then some of the other markers, like PALB1, had small mutations as well. So, but they don't seem to be concerned about that. But it is reported towards the end of the letter, and I'm what does that mean? My genetic counsellor hasn't really gone into detail. She applied the CanRisk algorithm to my personalised risk score for ovarian cancer, because obviously I've now been treated for breast, but I have a very significant risk of ovarian cancer as well. In 8-9 years' time, it starts to go up. And I was just thinking, so it wasn't a whole genome. They kind of did a targeted array, which kind of makes sense. Obviously, it's cheaper, and they were expecting to find this because of my family tree and how it looked when I was diagnosed with cancer. How does that work? So when you're actively working within the PBCB trial, it's really difficult for patients, of course, who didn't know that they had this germline mutation. But with the oncologist, you have to make a clinical decision because the sequencing takes still from the blood draw to analysis to, getting that data back. It took me about 6 weeks to receive it from diagnosis of cancer. But by that time, we didn't have time to waste because it was very aggressive, grade 3, and the oncologist wanted to start treating me straight away. So I started with platinum-based chemotherapy because she knew that it was likely that I would be BRCA positive because of the family history and also the age at which I was diagnosed, and that it was acting like a basal type cancer, which is more... Anyway, even without BRCA, apparently they respond better to platinum-based chemotherapy. So do you analyze the sequencing data for all patients who come from this VBCB trial? And then how effective is it at informing treatment for patients in real time?
Got it. So there's a lot of questions in the question that you've put to me, and it's really interesting to hear your perspective, actually. Okay, so there's a couple of parts to it, right? So when we do a clinical study, there is going to be the parameters within which we can act. And so the PBCP project, which is not one of my projects, is actually Jean Abraham and Carlos Caldas. They had the idea of doing whole genome sequencing for clinical patients recruited via the NHS and trying to use genomic information to inform treatment decisions to the best of their ability at the time. There's going to be a set of parameters that they will act under. However, if you recruit patients through the NHS, you also have to make sure that you don't compromise their standard clinical care. So it sounds to me like that you would have had the standard clinical care approach, which is a targeted panel for BRCA1, BRCA2. I think that's absolutely correct and exactly what would happen for anybody coming through the NHS in your situation. And I think those timelines are kind of about right. That is sort of what happens. I think that is actually a separate thing to the whole genome sequencing study, the PBCP study itself. That study was for us to learn and understand how to use whole genome sequencing. But the actual clinical piece and the sort of the clinical management piece was still coming through the standard process because we cannot compromise your standard clinical care, right? So I think that's why you got that twist panel. And today, if I see somebody with your family history in clinic, they're all going to get a twist panel. They're not going to get a whole genome. They're all going to get a twist panel today. Every single woman who comes in through the door in that position. The whole genome sequencing study was to a select group of patients who had cancer already. And when they do the sequencing, they sequence the cancer and the normal, the germline DNA. So, I guess in your question, you're kind of wondering, why did they do that panel? They could have just looked at the germline genome. They will do that too. Okay. But they were right to do the panel because they have to do it quickly and make sure you get the right clinical treatment. So that's why they did that piece. Then it comes to the study, right? Who analyzes that? So we are helping Jean to analyze it. That's actually only been quite recent. The sort of the earliest piece, there would be contracts through that sort of thing. And I think they kind of did that through Illumina and I think Genomic England, I'm not 100% sure actually, but we're now helping her to reanalyze it and use all our tools to try to give her the best information possible before she actually publishes that piece. So we're coming in kind of late really into that process. But the kudos has to go to people like Gina and Carlos who had the idea. When they saw that whole genome sequencing was producing outputs, they decided they were going to do that clinical study because that's the slow bit. The slow bit is starting it, getting all the ethics approvals, getting the study going. And it takes years to collect the patients and then to collect the treatments and outcomes. I'm coming in at the end to help her analyze right at the end. But when we're recruiting patients, we also have to make sure we treat them correctly. And that's why you would have had all the standard NHS pieces as well. So that's sort of trying to explain the chronology and why you will have thought, I got NHS stuff and I got research stuff and I'm not quite sure where it starts and where it ends. That's to answer that bit. Your next point, which I think is really relevant, and one of the reasons why I say it's a long process and it needs to involve a lot of people, you talked about the result and how actually nobody explained some of these parts to you, right? That is still a problem today. That's for a panel. That's for a panel in the NHS, never mind a whole genome. So the expertise and understanding required to be able to analyze, interpret, and report back effectively, that's still quite hard. So in the old days, the biggest hurdle was the sequencing because it was expensive. But now the sequencing is not expensive anymore. It's the analysis and interpretation. And you've just told me that analysis and interpretation of the panel was not ideal, really. They told me about the BRCA bit, but they didn't tell me about all the other bits. And I think we're still learning. I don't think there's any sort of malice in it or anything. I think people don't really know where to set the right cutoff. Do I put all of this information that could be useful at some point in the future? Do I put this into the report now, or do I hold it back and not put it into the report? And this is where science and medicine has, I wouldn't call it a gray zone. It is a learning zone. Because sometimes we don't know when we're seeing something, whether it is definitely the absolute and important genetic cause of somebody's disorder. So when I see a child with learning disability in clinic and I do a whole genome, I might find a mutation in a gene which has been reported before. And there's been reports of hundreds of different children with it. And I'll be able to go, yes, there's enough evidence out there for me to be sure that this is definitely the mutation that's causing your genetic disorder. But you can sometimes get a mutation in a new gene or a mutation that's never been reported before. And then you're kind of wondering, not sure whether this is important or not. So we end up we'll report, these are the definite mutations that we are sure is clinically important. Then there's a bunch of things here in this gray zone, in this learning zone, that we're not sure whether are important or not, but the question is whether you put it into the report or not, because it can be alarming to some people, right, if you don't have that understanding. And I think we're still learning that piece. I don't think we've got it right just yet.
Yeah. Thank you so much for that very comprehensive response. I now remember in hindsight, so this letter that I opened was my NHS diagnostic letter, which came a lot later than the PBCB results, actually. That came 3 months down the line, but the PBCB results were actually, I think that was, you're right, whole genome. But yeah, it's still, even as a scientist, especially when it's affecting you, you block certain things, and then you're like, okay, BRCA1, I'll just filter out the rest. And then when I saw that it was a twist panel that highlighted a few other small mutational signatures, I was like, oh, okay. A bit worrying, but if we still have, we're always learning, and the government has recently announced this £650 million investment into doing a whole genome sequencing of all newborns, I think by 2030, as part of the 10-year plan. What does that mean if we're kind of sequencing all these newborns, and what are we doing with the data? And from an ethical point of view, what does it mean to have your whole genome data stored, and who can access it? And what does this mean for the new generation? And what do they do with all of this data about themselves?
Another enormous question. So, if we look at where we are about learning from data today, we will not have made the leaps in progress in understanding if we didn't have access to a lot of the data. And I will say all the data that I've ever worked with has always been anonymized. They're very personalized. Every patient is so individual. But I don't need to know the person's name or date of birth or where they live. I just need to see their genomic data to understand and learn about it. And then I need to see that person's data in a sea of other 20,000 other people to understand those tumors behave like this, that set of tumors behave like that. That's the value of having all that data together. So I'm a big proponent of generating the data and sharing it centrally so that we can learn from it. And especially if you have a nationalized system like we have in the UK, where, you can link it up to hospital episode statistics, you can link it up to Office of National Statistics data and look at mortality and outcomes. The ability to learn from that is just enormous. So I think you must have spoken to Daniela at the PBCP meeting. She's the one who's analyzed the breast cancers across England, and her ability to look at the genomes, link it up to Office of National Statistics data, and show that there are some genomic features that are super important and some that are less important is really powerful. So I'm a big proponent of sharing data to the middle, especially for something like cancer, because cancer is so individual and there's so much information. Cancer genomic data is very different to whole genomes of newborns. That is your germline, your genetically inherited information. So those are different things, but equally important. And so in my clinic, I look at the germline, right, rather than the cancer. In my research world, I look at cancer, but in my clinic, I look at the germline. So in my clinic today, I will be recruiting patients who have a learning disability. We don't understand why they're, falling behind at school. They're trying to get support, and we'll be trying to make diagnoses. Before the advent of next generation sequencing, as I mentioned earlier, I think our diagnostic rate was only about 10%. Today, because of the ability to share data and put it to the middle, it's about 40%. It's a lot higher. So that's a good thing. So I'm all for that as well. I'm all for, if we don't understand, we want to try to look, see, and then share information so that we can try to figure things out. Because I might find a genetic change in a gene I've never seen before. But then in Manchester and in Newcastle, they might have seen the same thing. And now we together put our patients together and go, look, they look the same. They have the same clinical phenotype. It's the same gene. It's in the same domain of a protein in the gene. And someone might do some experiments and show it's actually causative. And that's how we have our knowledge has increased and diagnostic rates have improved so dramatically. So there's a lot of power in that. Now, the UK is unusual. We have a nationalized system, so we can benefit from big national studies that start off as research and then eventually become clinical implementation. It happens quickly. There are problems with that too. But in general, it's a very well-meaning thing. And I guess the whole genome sequencing newborn thing is part of that, where they want a big research idea and then they want to show that it is beneficial and eventually gets to the point of clinical utility. And I'm sure that that's what's sort of driving that idea. So there's a lot of reasons in terms of learning and understanding disease that whole genome sequencing can be very powerful, or genome sequencing of any sort doesn't have to be whole genome. But I absolutely agree that we do need to balance that with a patient's understanding of what it is we're doing with that data and then how that data is managed and stored. And these are really important things for us to get right, because people are very concerned about privacy and they're very concerned about companies getting access to their data and, issues with insurance and this sort of thing. And then people also mistake, because the word data means many different things to different people. Some people worry about this sort of genetic data being misused, the way, people talk about Cambridge Analytica and Facebook and all, this sort of data as well. That's a very different kind of data. The whole genome data that is being generated from patients from research studies is always anonymized. So we don't have your names and dates of births. It's somewhere central or locked down. You have to have the right credentials to access it. You have to abide by all these principles. There are regulatory teams that check on your projects. You have to provide reviews and updates every year. So there's a lot of pieces in place to ensure that your data is protected and your data is looked after. In fact, when I am looking at whole genome data in the research environment in Genomics England, which is where a lot of this data is stored, I can't tell which one is my patient because they're all so anonymized that I just can't tell. And that's the way it should be. So I think that's a good thing. And I think what we need to do as a body of people who are in the genetics genomics field is to actually actively try to educate the population about what it really means to share your data. So, a lot of people are so worried about data sharing. But actually, if we're talking about anonymized data, which are numbers and letters that nobody really understands, it's okay. It doesn't really matter, right? If I lost my genome, if I put my genome on a stick and I lost it on the train, I don't think it's going to make very much impact, actually. A lot of people kind of go, well, your genome is very personal. It's very individual to you. And that's true. But it's not very easy to interpret, to analyze and to interpret. And even if you did, there's not very much in your genome that is very predictive, actually. It tells you about risk, but it does not tell you about fate. It's not like, you might have a mutation in BRCA1 or 2, which increases your risk of cancer, but it doesn't mean you will definitely develop cancer, right? There's very few genetic disorders where if you have a mutation, like you almost certainly will have the disease. And that's something like Huntington's disease.
Right.
And actually that is the only genetic disorder where insurance companies can actually penalize you on only if you ask for life insurance exceeding £500,000. Today, insurance companies cannot ask you about your genetic diagnosis. They cannot ask you for a genetic test result. And they have agreed, this is their moratorium. They have together, the Association of British Insurers have agreed that they will not ask you for a genetic diagnosis. And so actually we don't have to worry about that component. Insurance companies use your family history usually. So your premiums are decided by your family history regardless of your genetic state. So actually that is a misconception that people worry about insurance too much. And I think what we need to do is to educate the public about what are the benefits of actually sharing your information like medical data or genomic data. They need to understand what is in place to protect them too, and that we are taking it seriously. I think all those things are incredibly important. But I think we also have a responsibility of using the right terminology, right?
Yeah.
Data is a big word, covers a lot of things. And then I think the last point you were making there was about children. Because when we are, as adults, make a decision about our genomic data as consenting adults, it's a very different situation to a newborn who cannot make a decision. But we've made that decision for them. And what happens to their understanding and their rights and what they want in the future. I'm going to say I don't know enough about that sort of angle, but I think there are a lot of people who are trying to defend that or argue about it and discuss it rather to try to find the right path. Yeah, so I think it's a interesting set of questions, and a very interesting one for us to look and see how that changes. And I think that'll be slightly different for each country. So I can already see that even for the cancer sequencing data. So it'll be an interesting one to watch.
Absolutely. Thank you so much. So we've spoken about all these kind of de-identified, anonymized genome data, which is like a series of letters and numbers. How can this, at the population level, because you've looked at thousands and thousands of these signatures, can you tell us any stories of how this has made an impact or difference at an individual level to any of your patients?
So when you have that de-identified or anonymized data at scale, you can understand what are features that are important, which features could tell you whether someone is sensitive to a drug or could tell you who might do well or who might do badly, right? So a good example of that one, we've had a couple. One's been in Cambridge and one's been elsewhere. So a patient in Hull who actually had something called an angiosarcoma. This is a cancer of a blood vessel wall lining, which is a very rare cancer type. But today, if you have a cancer, you get treated based on the organ of origin. So if you have a breast cancer, you'll be treated for breast cancer. If you have a colon cancer treatment. Angiosarcoma treatment. And he didn't respond to any of the standard angiosarcoma treatments. And he was 30 years old, he was a young man, and he had a genetic disorder anyway. And he had sort of the first treatments and unfortunately returned sort of within the year with evidence of metastatic disease. And his disease was very aggressive, so he progressed very, very quickly. He had metastatic disease everywhere by July 2018. And we were asked to do his whole genome. And remember, at that time, whole genomes was still very much in the research space. It was not in the clinical domain as it is now. And treating somebody on the basis of a whole genome absolutely hadn't been done before. But here's a 30-year-old. He's got disseminated metastatic cancer. He's not responding to anything. So we do his whole genome and we find that in his tumor, he's got a signature, a mutational pattern that tells us that he must have an abnormality in a gene called polymerase epsilon, POL-E. We hadn't found a polymerase epsilon mutation, but we could see the signature. Now, it's not work by us, it's work by other people in the US. They have shown that if you have a polymerase epsilon mutated cancer, you're very sensitive to immune checkpoint therapies. At that point, immune checkpoint therapies hadn't really been approved for cancer use in the UK necessarily. But here we were with a 30-year-old, and we found that he had the signature. Eventually, we found a polymerase epsilon mutation. It was missed by the initial analysis, and we had to do a lot of additional studies that provide the evidence that this might be a tumor that would be sensitive to immunotherapies. But because it hadn't really been approved, because that's the process, right? You have to do all the clinical studies to then get approval from NICE before you can use a drug. And whole genome sequencing hadn't been through that process. Immune checkpoint therapies hadn't been through that process. So we had found this, we believed this man to be sensitive to this drug, but we couldn't give it to him basically. But we had learned it on the prior, because we had studied all those other cancers and somebody else had already done the work to show that polymerase epsilon cancers were sensitive to immunotherapy. So there was a lot of prior evidence. We eventually managed to find somebody who would give him the drug, but NHS wouldn't pay for it. I've never seen so much courage. I'm always so inspired. And this is that ongoing theme. I'm kind of more inspired by the patient sometimes these days than anything else. And he was so brave, and he just wanted to give it a shot. He says, I've got nothing else to lose. I want to do this. He raised money. He crowdsourced. It was incredible to watch the community come up around him in Hull. He was in the newspapers. They had a black tie ball. They did all sorts of things in the community to raise money for him. And then he gets his first dose in London, and then he moves hospital to get his remaining doses. But the point when he is receiving the immune checkpoint therapy, he's so ill. He had mets in his face was kind of deformed, and he had mets everywhere. His lungs were collapsing all the time. He couldn't leave the hospital. And by the third cycle, everything is melting away. Wow. By 7 cycles, everything's melted away. This man walked home. This was 2018. He's still alive today. So that was a real sort of moment for my team and I of realizing this works. We could actually drive this message home and try to sort of show that mutational signatures could be used to really inform clinical decisions. And it can be based on all that data that has been generated in the past and then used, and all the information about, outcomes and, Office of National Statistics, etc. We can use all that information effectively. So that was for us the point where we thought, and that was for me the point where I went, I'm going to do academia as a career now, because I think this is going to be beneficial. But I think the hurdle now, the understanding and the learning is really important for us. But that next bit, the clinical studies, getting the evidence for the clinicians, I don't blame the clinicians that they're going, I'm not sure how to use this information. I think they're right, and what we need to do is get them the evidence base to support them so that they feel that they will be supported if they wanted to give a patient a treatment that was unusual.
Absolutely.
But that shift in mindset is, and it will require a shift in mindset, is that we shouldn't just be treating patients on the basis of what tumor type it is, and that we should be looking at the genomes, in some tumor types especially, looking at those genomes and trying to inform the treatment plan with that personalized information from each person's genome.
Absolutely. So what's the one key takeaway message that you want to leave our listeners with?
Academics, we'll have our agendas, right? We're gonna want to do certain things in our careers. But I think the voice of the patient has to be one of the loudest ones. And actually all the success stories that we have had have been largely driven by the patients going, but I want this, And while we need to collect that evidence, the evidence from clinical studies to inform the decision. So as you say, getting the big data, getting all that data anonymized, etc., and learning about it. I think my plea to people, if you are invited to participate in one of these, your data is protected. We don't need to know who you are. We just need to know what is in the cancer and how to use it. And we can use it to help many people across the country, many people across the world. So I think that my first point is don't be afraid of the data. The data is safe and we can use it without knowing who you are. And then the second thing is that this is probably for the clinicians and for the academics. We collect data at population scale and in big studies to get p-values, to get statistical significance. But the person you are treating is an individual. And when you are sitting opposite that person and looking at their data and their genomes and everything else, you are treating a person. And the background knowledge that you're leaning on is just helping you make that treatment decision. We still have to think. We cannot just go with an algorithm, right? We have to be able to think about each person and go, what is the best path for this person? I think it's going to be an interesting 5 to 10 years. I think we're going to see how people think about data and use it. They're going to see that change. I think the UK has been a bit of a leader in doing those studies to make it permissive for us to make that mindset shift. But the actual using the information to make treatment decisions, that big clinical piece hasn't quite happened yet. PBCP is a nice first step. I think we need to do something systematic across the country to show people that you can benefit, anybody and everybody can benefit from this sort of study, and your data is protected and it's all good, but we can actually make personalized medicine a reality.
Amazing. Thank you so much, Serena.
Pleasure.
It's very rare to see like academic research making actual impact for patients, and it's super inspiring to hear, you know, just how much you guys are doing to ensure that this is a reality and that it starts to affect even more people in a positive way. So yeah, I look forward to seeing what more is to come from your work.
Thank you so much, Asin. Thank you so much for the invitation to come and chat today. It's been really nice to sort of talk about stuff that's been going on in the past. And I think, you know, in the end, we can't do these things without people like you sort of, you know, championing it and making it accessible to the general population. So thank you.